And if we understand these pathways and the complexity of these pathways and what the hell is going on, we can then use that information to overcome the apoptosis resistance. That is, we resensitize cancer cells for apoptosis. And that is again by using the information we gleaned from here. That is, in this case, we target the PI3 kinase signaling cascade. However, should you have any other ideas, or should you find any other interesting targets, there's actually a web page by the American government, clinicaltrials.gov, where you can look whether there are clinical trials on the pathway you find interesting. So either you can, if you wish, stop the video now, or after the lecture go to this web page and type in the cancer that interests you and the pathway that interests you, and you can look whether there are clinical trials ongoing. It also gives you a good information about the pharmacological inhibitors used in those clinical trials. It's a good place to play around and glean extra information. And this basically brings us to the concept we work in, the concept I like to use in the context of the PI3 kinase pathway, and that's the inducer and sensitizer concept. And this is basically, we use a treatment that induces apoptosis, that would be chemotherapy or radiotherapy, which usually, because cancer cells are resistant towards apoptosis, doesn't kill all or even most cancer cells. It would kill all cancer cells if we increased the concentration or the dosage of therapy, but that would also cause more side effects and eventually kill the patient. So we are limited by not killing the patient. Now, we take a second component, or third, fourth, and fifth, that sensitizes the cancer cells for apoptosis. So we use either an inhibitor, an antibody, or a vector that takes away the alteration in the cell which makes the cancer cell more resistant towards therapy. So we sensitize the cancer cell again for apoptosis. Importantly, because that change in the cancer cell is specific to the cancer cell, that sensitizer often has very little effect on the patient. It, however, has very little effect on the cancer cell on its own, often, because you take away the reason why the cell doesn't die. However, on its own, you don't give the cell a reason to die. What you often get, or sometimes get, is that the cancer cells stop proliferating, which on its own is already an achievement. Now, if you combine those two treatment arms, you get a combination where you either increase the amount of cancer cells dying via chemo or radiotherapy, or you can use less radio or chemotherapy, therefore reducing the side effects, and still kill an equal amount of cancer cells. So basically, the idea is use less stress on the patient by killing equal or more cancer cells. So either increasing the quantity of life or the quality of life, or both. Okay? And we'll look at an example of this using the PI3 kinase AKT-mTOR survival pathway. Now that doesn't come as a big surprise after I harped on about it for ages. And we'll use neuroblastoma as an example. Neuroblastoma is a fairly interesting childhood cancer. Low stages of glioblastoma neuroblastoma can actually spontaneously regress. stage 4 neuroblastoma is a particularly nasty disease which has to be treated with very strong chemotherapy with lots of side effects. It is also fairly common in very small children. It even occurs when children are born that they are already born with neuroblastoma. So it's a disease I'd like to get rid of as quickly and as humanely as possible, because I don't want babies to suffer, basically, not surprisingly. Now, interesting, neuroblastoma is frequently characterized by inactivation of caspase 8. So the promoter of caspase 8 is methylated. Neuroblastoma cells don't express caspase 8, so targeting the death receptor by TRAIL or CD95 or TNF-alpha doesn't really work with neuroblastoma. Okay, now we stop the video and we will think about the treatment for neuroblastoma. So I want to treat neuroblastoma with chemotherapy, and I want to sensitize this tumor by blocking the PI3 kinase AKT-mTOR signaling cascade. It is actually very little is known about this cascade in neuroblastoma, so there are no obvious mutations, but we know that it is highly activated. So it is a good target. We know that in neuroblastoma we have increased expression of receptors. So activation occurs early on. Already here we have more active PI3 kinase, and that goes through the whole pathway. And I would like you to think about at which stage of this pathway would you consider blocking it for combination therapy. So do you think PI3 kinase is a good target? Do you think AKT is a good target? Do you think mTOR is a good target? There's no right or wrong. And basically, the idea I had turned out to be at least slightly wrong. But what target would you think is a good idea to combine with chemotherapy? Where to put the block on this cascade to enhance the killing effect to get rid of neuroblastoma as quickly as possible? Good. So what we did is we used a dual inhibitor, which we generally call BES. That is an inhibitor which blocks both PI3 kinase and mTOR. So it targets both. We used as a readout phosphorylation of AKT as an indirect target of PI3 kinase, and phosphorylation of S6 as an indirect target of mTOR signaling. And we could find that basically within 1 hour, or if you look at S6, within 4 hours, we got an inhibition of phosphorylation. That means the inhibitor on our neuroblastoma cells worked within a few hours. If we looked at the cell cycle distribution, again remember our cell cycle, this is G1, S phase, G2, that should be G1, G0. We see a statistically significant alteration maybe after 6 hours, but one which we would call convincing after 12 hours. So 12 hours after giving an inhibitor for PI3 kinase and mTOR, we see that the cells accumulate in G0, G1 phase. Therefore, we believe that we have an effect on proliferation of these cells within the first 12 hours. What we did next is we used the combination of inhibitor and chemotherapy, in this case doxorubicin, and we did 3 experiments. We gave the inhibitor 12 hours before we gave the chemotherapy. We gave the inhibitor at the same time as we gave the chemotherapy. And we gave the inhibitor 12 hours after we gave the chemotherapy. Now importantly, chemotherapy, which is the red arrow, was always given to the cells for the same amount of time. So the inducer of apoptosis was always present for the same amount of time. The sensitizer for apoptosis was either given 12 hours before we induce apoptosis, at the same time we induce apoptosis, or 12 hours after we induce apoptosis. If you think about the clinical context, what you normally do is you give the inhibitor prior to the inducer. You make sure by testing plasma levels that you have the maximum amount of inhibitor present, and then you induce apoptosis. Now, if you now look at figure A down here, where we have the amount of sub G1 cells, the percentage of cells which have less than normal DNA content, where we have fragmented DNA and therefore apoptotic cells, what you find is that in the presence of the inhibitor, we actually get, at this concentration, less apoptosis. If we give the inhibitor at the same time, at the high concentration of chemotherapy, we get an increase in apoptosis, what we would expect. However, if we give the inhibitor at only the last phase of apoptosis, we get a huge increase in apoptosis. So basically, we inhibit the PS3 kinase signaling cascade at a very late stage of apoptosis, the last 12 hours. We get more apoptosis, and by inhibiting a survival pathway for the longest possible time, we actually prevent apoptosis. Now, how does that make sense? We take away the survival signal for the longest time, and therefore we block apoptosis. That's fairly counterintuitive. Doesn't make any sense whatsoever. It wasn't the result I expected. And that's basically going back to our slightly complex slide of the role of PS3 kinase signaling and why I asked you at which stage you would inhibit the signaling cascade. You have to consider that PS3 kinase signaling is not only a survival signal. While regulating cell death is a central aspect of this survival cascade, it also has other roles. It regulates cell proliferation, it regulates the metabolism of the cell, and it also regulates cell motility and the cell's response to DNA damage. So basically everyone calls it a survival cascade because its most obvious role is in regulating the cell survival, but it regulates many different aspects of the cell. And it regulates these aspects at different levels and at different speeds. So if I inhibit PS3 kinase, the effect on the metabolism might be visible before the effect on survival. And that metabolism effect can then affect survival or cell death. And what we saw here is that in neuroblastoma, the inhibition of PS3 kinase was important at the level of the mitochondria. So basically we inhibited PS3 kinase, therefore AKT was inhibited, and the cell death we saw was regulated at probably this level, because we only needed the 12 hours. The most important part of the cell death was down here, where AKT blocked GSK3 beta. The blockage of GSK beta, and we could show that by using an inhibitor which is specific for GSK beta, that led to even more apoptosis. So basically what is happening, inhibition of PS3 kinase is important for the sensitization at the level of the mitochondria that we checked, but more importantly, it is also the role of AKT being blocked, which in turn activates. AKT is blocked, so GSK3 beta is activated, and that leads to even more apoptosis. So, what happens if you treat neuroblastoma cells with doxorubicin? You get a certain amount of apoptosis. That apoptosis is also mediated by the mitochondria, as we discussed earlier. Doxorubicin is a chemotherapy. That means it goes via the mitochondrial pathway. We get DNA damage via topoisomerase 1 inhibition. Then we get release of cytochrome C from the mitochondria via pores formed by the BCL2 family. Now, if we now add the inhibitor of PS3 kinase signaling, we inhibit AKT, which normally inhibits GSK3 beta. So we now have an activation of GSK3 beta, which phosphorylates VDAC. VDAC is able to form pores with the BCL2 family, and so we get an increased release of cytochrome C. So basically, by inhibiting PS3 kinase signaling, the important event which occurs in neuroblastoma cells is we get increased destruction, if you want, of the mitochondria. We get increased release of components important for apoptosis at the level of mitochondria. And that's sufficient for the inhibition to occur late on. So basically what we have again is we prepare the cells, or we damage the DNA in the cells with doxorubicin. We induce stress within the cells, but the cells resist the stress. They do not commit suicide because at the level of the mitochondria, the signal isn't strong enough. Now we have built up the stress at the mitochondria, we take away the brake, and then fairly rapidly, that phosphorylation event is within 15 minutes, we get the effect via VDAC and induce apoptosis. So we only need about 15 minutes. We need a very small window where we stop PS3 kinase signaling. Now, if we use the pre-treatment where we get a reduction of apoptosis, which is what we don't want, that's probably due to the cell cycle. Basically what we do here is we already block the cells from proliferating, from cell cycle progression, prior to giving doxorubicin. So those cells, 12 hours, are already not cycling. That means they're not in S phase. We've got an increase of cells in the G1, G0 phase. And those cells, if we damage the DNA there, are less responsive because the DNA damage is most important in proliferating cells. So here, by inhibiting survival signaling, we accidentally also inhibit proliferation, and we inhibit proliferation with pre-treatment too long, and these cells become more resistant. We also have the induction of autophagy during the whole event. Now, that's probably regulated via the PS3 kinase mTOR route, so some of you might have picked AKT as a good target. Now, interestingly enough, there's a publication which suggests that if you hyperactivate AKT, you induce huge amounts of cellular stress and at least stop the cancer cells from proliferating. So again, we have a problem here. We have something which we call a survival pathway, but depending on the very stage of where we inhibit or activate the survival pathway, we can actually get results that we don't want. So we have to be very careful in combination therapy how we combine the effects of the inducer of apoptosis and the sensitizer, because otherwise our sensitizer very quickly can become a blocker. Okay, now we are getting towards the end of the lectures. I hope you're all still awake, and for the last couple of slides I would like to talk about a form of apoptosis, it is caspase dependent, called anoikis. Anoikis is also ancient Greek. We have now learned that we use ancient Greek to sound intelligent, and it means homelessness. So basically a cell commits suicide because it's lost. It loses contact with its environment, it doesn't know what it's doing, and therefore it goes, I'm lonely, I will die. Now, we believe that anoikis is an important aspect if we talk about adhesion-mediated apoptosis resistance. So a cell, depending on the context it exists in, can become more resistant towards apoptosis. Cells interacting with other cells are less likely to die than cells which are not interacting with other cells or with the extracellular environment. And this is something which has been known, again, since the 70s. It's been shown that cells, cancer cells in different forms of interaction are more or less resistant. So since the 70s, we've known that the mutation of the cancer alone doesn't determine the resistance towards apoptosis. It is also the microenvironment around the cancer cell which influences the resistance. And that's something we termed AMA, or adhesion-mediated apoptosis, or anoikis resistance. So the cell sticking to something becomes more resistant towards apoptosis. And traditionally, people refer to this as CAMDR and CAMRR, which is cell adhesion-mediated drug resistance or cell adhesion-mediated radioresistance. And we just decided we just call it AMR because it's friendlier and it sort of composes those two aspects. And basically, what you have in a cancer cell or any cell, you have two forms of interaction. Either a cell can interact with another cell via, for example, connexin, or via beta-catenin mediated interactions, or cells can interact with a substrate. That means with extracellular components, the extracellular matrix. And that interaction is often mediated via integrins. And again, you'll see we have lots of old friends, as we now call them, old friends which are again activated or inhibited via those interactions. So for example, you have here beta-catenin, you have PS3 kinase signaling, leading to ERG, leading to increased survival. With integrins, you have our old friend SARC, you have FAC as well, and you again have PS3 kinase signaling leading to increased survival. So those interaction points induce signals, even if their pathways are not mutated, within cancer cells that enhance the survival of cancer cells. Now, if we take away those interactions, those cancer cells either become more sensitive to our treatment or can be induced to commit anoikis. Basically, we let the cancer cell think it's completely on its own, not in the body anymore, and it is blindfolded, and then it dies. And this strategy in our lab we have termed Alcatraz strategy, because we are scientists and have a childish sense of humor. Basically, if you think about it, Alcatraz is again symbolic for taking away the connection of the individual prisoners. So you have a prisoner in a cell being disconnected from the prisoner in the next cell, hopefully. And then again, you have the whole prison being disconnected from the mainland via the ocean, which is full of sharks. So basically, Alcatraz, or the Alcatraz strategy, is taking away as much of the environmental cues, putting the prisoner or the cancer cell into isolation. And that works. However, you have to be fairly careful. You have to think about what happens if you take away interactions. Because interactions are not just signaling of making sure the environment is still there, but they are also adhesion points. So that makes the cell more sticky. So if you take away the wrong adhesion of a cancer cell, what do you think can happen? And there's a hint on the screen, and we'll give you a couple of moments, stop the video, to think about this. So what can happen is that the cells become more adherent. So if you pick the wrong interaction to block, the wrong cell-cell interaction, you might make the tumor cell think that it's more alone than it actually is, but you also might enhance its motility. So basically, you cause metastasis, or you can induce a more metastatic phenotype. So just randomly blocking cell-cell interactions or cell-substrate interactions, not a good idea. Pick your targets very carefully. For the next couple of minutes, we'll show you some movies where we did exactly that. We carefully selected contact points of cells, inhibited those, and I hope I convinced you that that is a really good strategy. So we'll see. Okay, what we have here are glioblastoma cells. That's a fairly common brain tumor. These cells are highly motile. This is a time-lapse image. It's 48 hours compressed to about 40 seconds. These cells, you can watch them move. They're very fast and they are highly invasive. That means if you have a small tumor on the one side of your brain, those cells will be already migrating to the other side of your brain, making it hard to get rid of them surgically. Okay, so those are completely untreated cells. Okay, now these are the same cells treated with an inhibitor of cell-cell interaction. Now I could tell you that you see a difference here, but to be honest, had we switched the films around, they would look completely identical to you. That's not particularly surprising because these cells are mainly moving along on plastic. They are actually not interacting with each other. So these cells are slightly slower, but basically I don't expect you to see a big difference. Okay, good. Okay, now these cells are treated with a substance which stops their interaction with the substrate. In this case, because it's an experimental plastic, but it works the same for fibronectin or collagen. Now you'll see a very limited effect first, but if you focus on the lower left side, you will see that these cells move now as one. So they interact more with each other. The adhesion to the floor goes away, cell-cell interaction is enhanced, and about 40 hours after treatment, they start dying. These are cells treated with both substances at the same time. Here the cells detach much quicker because they can't interact with each other to block this detachment, and already within 3 hours of treatment they begin to die. And what you now have here are nice classical apoptotic cells. You see membrane rupture, not membrane rupture, but membrane blebbing, dying cells, detached cells, anoikic cells. So combi treatment enhances this effect tremendously.